Interphase. This is the start of the cell cycle, where G1, S, and G2 occur. G1 is when the cell synthesis mRNA and proteins. S phase is when the DNA is perfectly synthesized and replicated. The only abnormality would be mutation. The G2, the rapid growth and increase of protein synthesis to start meiosis. Meiosis 1. The first mitotic division is reduction division, diploid to haploid, in which homologous chromosomes are separated. Chromosomes in prophase 1, chromosomes condense, nuclear membrane dissolves, homologous chromosomes form, bivalence crossing over occurs. In metaphase 1, the spindle fibers from opposing centromes connect to bivalents at centromeres and align them along the middle of the cell.
In anaphase 1, spindle fibers contract and split the bivalent homologous chromosomes move to opposite poles of the cell. In telophase 1, chromosomes decondense, the nuclear membrane may reform, and the cell divides through cytokinesis to form two haploid daughter cells. Cytokinesis is the splitting of the one cell into two by way of cytoplasmic division. And interkinesis is not a part of meiosis. This is a resting phase for the cell cycle where DNA is not replicated.
So then the start of meiosis 2 begins with the second division, which separates the sister chromatids. These chromatids may not be identical due to crossing over in prophase 1. Prophase 2. Chromosomes condense, the nuclear membrane dissolves, and centromeres move to opposite ends, perpendicular to before. Metaphase 2. Spindle fibers from opposing centrosomes attach to chromosomes at the centromere and align them along the cell equator. In anaphase 2, spindle fibers contract and separate the sister chromatids. Chromatids, now called chromosomes, move to opposite poles. Telophase 2. The chromosomes decondense, the nuclear membrane reforms, cells divide with cytokinesis to form four haploid daughter cells.